This tutorial will be focused on how you can turn a single brain section into a quantified results where all the neurons are segmented and registered to the Allen Brain Atlas, and you can then plot it as a bar graph, for example. So we're going to use RStudio here, and we have an example section here, which is a folder that contains multiple TIFF files. And these TIFF files are each not that large. Each TIFF file is covering one part of the brain section, and each TIFF file, as we can see here, is 2048 by 2048 pixels, and it's a 16-bit raw TIFF. And in general, this is the file format, 16-bit raw TIFF, that WholeBrain is using. So we're going to start loading the WholeBrain package here. I'm just going to clear the console, and then we're going to start by extracting all the images in this folder. So we start by first defining our folder path here into an arbitrary object we're going to call folder. And then we're going to extract the images that are in this folder. And in WholeBrain package we have the command from this, which is called get.images, and it takes as input a folder. And if we let's see, yeah, it takes an input as x here is a folder, the full path, and type is default TIFF, but it can really be any image format you want, JPEG or PNG or whatever you want to look. Default is TIFF. So if we just write here, it's going to look for TIFF files in, and we assign all of these images to an object called images, which is a character vector. So here we see 63 different entries, and it contains the full path to this image here, as we can see. Okay, so we can check the length of this character vector images, and that will give us the number of images we have here in the folder, and it's 63 image tiles. So let's write some comment sections to the code as well, so remember what we have done. So if I use the hashtag here, that code will not run in the console when I then enter it. So now we have the code we did here. And we can also comment here that this sets the folder path, so we don't remember what this part of the code is doing. Okay. Now, if we would start here now, what we actually want to do is to stitch all of these sections together, or rather all of these image tiles together to a single brain section, which we have imaged with the microscope. So to do that, WholeBrain has a command that's called simply stitch. But in order to understand what the stitch command is doing, I'm going to show you a command that's part of the stitch pipeline, which is called getGridCoordinates. It takes as input just the number of images you have, and then the tile size, and then the overlap between the tiles, because normally it's common to have the tiles overlapping partly. And then merging, so you don't get any artifacts from like small mechanical vibrations when you do the imaging. So we set the plot grid equal to true in order to see the grid coordinates, and then we set 1 to the length of the number of images we basically have, which will create a numerical vector that's just 1 to the number 63, and the tile size was 2048 pixels. Now this is enough, so we'll run this like so. Okay, and this outputs a plot here. Let me just blow this up a little bit larger. There. Okay, so as you can see here, each number here is a single tile, 1, 2, 3, 4, 5, and they are lined and overlapping at the edges. So you can basically see this 0.1 overlap, which is 10%, and you can also see that we roughly have like an image that will be 13,000 times 16,000 pixels. So that's basically how the stitch algorithm is going to stitch all of these tiles together now. And I mean the order can differ depending on your microscope. So there are settings if depending on like how your microscope is moving up and down in order to take this image tile. Some microscopes moves just as we read from left to right and down, whereas other moves rather the opposite from the bottom and then moves left in a snake fashion like this. So you have this kind of option like either row by row or snake by row, and either right and down or left and up, and this kind of option you can set, which is basically different commands to the stitch, either the order command or the type command, where type is, for example, snake is the default and order is the default left and up. But if you would have other settings, those are available as well. So okay, let's get started with the stitch command then. So we write stitch, and since the default is what we're going to use, which is left and up, is the order, and the type is snake by row, which means that we start in the bottom right corner and it moves left and then up in a snake fashion. So we're going to take the folder path as input to the stitch. It can also take all the image vector, but we can simply provide a folder and it will extract images for us. So we actually don't need, well, yeah, we want to display the image. So by default show image is false, but if we just want to display one image, we can just run it now here, and it already loads all the tiles now into RAM, and now it has started doing the stitching, and now it's saving the image, so it's already stitched and done, blended in the seams and everything. So we can see here now it has created a new folder with the prefix stitched underline and then our section 001, and inside here we have the TIFF image, and this is the TIFF image now, the fully stitched image, and it displays it by default just the raw 16-bit, so it's very dark, but you maybe can see that there is a little bit of square grid pattern going on here. So that's actually the next step that we will try to see what we can do in order to remove this vignetting effect at the end of the tiles. So in order to display this vignetting effect at the edges of the tile, I'm going to show you a very useful command, which is simply called image show command, or imshow, which just takes the image and display it for you, and you can then adjust the brightness and the contrast for yourself. So this is really good for visually inspecting results very quickly and just looking at images, nothing else. So the imshow command takes as input this file name, which I just copy here into a new object called file name, which will contain the full path to this stitched section that I want to display now. So I run that, and now I run the imshow command with my file name. So like that, and then we run this. There. So it starts loading now this roughly 300 megabyte TIFF, and now it's open. So there we have it. And we also get this control bar that you will recognize later on when we do the segmentation. And as you can see, the control bar, you can then adjust the upper and the lower limits that you want to display. It's called like an 8-bit render, because your computer screen would roughly only display the 8 bits, whereas your image format is 16 bits. That is 2 to the power of 16. Okay. So you can see that what we essentially will do with the segmentation is trying to find a range where we have the fluorescent signal that we call neurons, right? And you can see that it's roughly somewhere between here, like sort of, so maybe 6,000, 20,000, something like that. In that range we have real radius GFP signal. So we're going to look for neurons in this range. So this you roughly set just once per experiment. And then also you need to provide where we're going to look for the autofluorescence, which we will use in order to find the brain section. There, right? There now I really restrict the range. You can see that you can really find an isolated range where you have only the autofluorescence of the brain section, so you can find that as well. So that's roughly the idea behind the segmentation. But yeah, we also have the zoom feature in the image show. So if I click on the image, I can see the overview of the image, but I also see a zoomed in part of the image. So yeah, and as you adjust, you see both the zoom in and the image changes. And here you perfectly see in this range that we have this really grid shaped pattern, the vignetting effect of the tiles, and we're going to remove this by a command called flat field correction, which is simply taking averaging over all the tiles and smoothing and then adjusting for this vignetting effect. So let's start the flat field correction now. The flat field correction is a simple command. It's just flat field correction like this, and it provides similar like Stitch. It provides you have to provide all the tiles as either like a string of or character vector of all the images. Now we run it, so it has loaded all the image in RAM, and now it's generating this correction image that it will then subtract and or actually divide from all the image tiles, so we don't get this vignetting effect. So now it's running the actual correction, and we can see this is the gain image, which is essentially the image that's used for doing the correction. So if there would be anything weirdness, you can open that one and see the results. We also get all the results now is displayed here in the flat field correction folder, which contains an equivalent number of image tiles. They have just now been flat field corrected. But in order to see the result, we need to stitch this together. So we then apply the stitch command, but to the folder where we have the flat field corrected tiles, which is essentially this folder, similar folder as the section are, only that it has the prefix FFC there, right? So we're going to run that and use this as input to the stitch command, just as we did before, and then we stitch again. So it's reading the tiles again into memory here, and now it's stitching and blending at the seams. Okay, now it's saving to disk this stitched image. Okay, we can see, yeah, we have a new folder. Yeah, it's done. We have a new folder here now with stitched underline FFC section, which is essentially the file I'm going to display to you now using the image show command, so we can see the difference what a flat field correction actually do to your image results. So we just add the FFC appendix here, no prefix. Yeah, like that, and then image show on the file name. Okay. Oh, we got an error. File not found. Oh yeah. Oh, the other way around. I've written the file name wrong. It should of course be stitched underline FFC underline your section name, since it's doing the stitching last. Okay, now we can run. Yeah, and it finds this file. So now it's loading this and will display it for us. So now it's here. Let me just open it there. And let me just drag the, yeah. And you can hide the zoom by pressing Z. It will hide the zoom, and if you click on the image, it will appear again. So you can see now that we don't have this vignetting effect at all. Can be other, like a little bit small artifact, but of this whole procedure, but essentially we get a smooth, nice image with no tile vignetting effect. So yeah, that's is about stitching and flat field correction. So now we will look more at the segmentation side of it. So I already said that the segmentation had a lot in common with the imshow command, but you have just more options, and it's more inclined toward actually producing some kind of like output, whereas the imshow is simply just to inspect images. So for the segment command, it takes as input also already stitched image tile of an entire brain section, and we're going to save the output of the segment command into an object which we just call seg. We can call it anything really, but and we take that input our flat field corrected image. And the input parameters to the segment command is both the number of sort of binary thresholds we want to use, which is 8. Resizing, if you have really huge images, by default, in order to display it on screen, it resizes about a fourth. And then it has a display option, which is by default true, but you can actually segment without actually showing the result. You can provide the parameters already by the filter, which will make more sense. I'm also going to put the ACO command to false, and that will make more sense later when we look at the registration. But essentially, the ACO command cleans up all the results a little bit, and I want to put it at false because I want to show to you what you do if you don't get desirable results as you want with the registration. So we start the segment command here now, and it essentially works like ImShow. You just get much more options in order to control. So we have a histogram here as well, where we can see the intensity range of the image. We see that we have a majority of the pixels are black, which is, and then we see the autofluorescence of the brain section, and then we have this huge range with the neurons. So the first part of a segmentation, binary segmentation, you would actually set the 8-bit render, because this is sort of how all the results later on are going to be superimposed on this 8-bit result, which is for the web format is going to be rendered in JPEG, and also for the plots. Then you can move around and see that we already seems to have gotten a suggestion from the computer itself, but it's not like entirely super good. But it seems to find sort of where the fluorescent range we have neurons. But as you can see, there's a lot of things that are not. Yeah, in order to show and hide the segmentation result, you just simply press H, and this I'm doing in real time, so you can simply swap between the raw data and your segmented results. You can see for yourself how the result looks. So it's important to set, yeah, you see here we have a huge blob, which is clearly too large to be in a cell. So these kind of things we're going to set with parameters. And then again, you normally set this maximum once per brain, usually once per experiment. It has to do with your imaging parameters and your like perfusion results and stuff like that, autofluorescence. But we start in the other corner when we have set the 8-bit range. We then set resize here, and I set it to roughly sort of like 70, 60, and that's that's the size of a pixel. So the size of a pixel here is sort of 0.7 or something like that micrometer. So you set the size of a pixel, and then the brain outline is basically where in the fluorescent intensity we find thresholding the brain outline. As you see, when I put it up to 900 here, you can basically see now that we have this contour that goes by the brain, this blue contour line, and that means we have succeeded in finding the brain. We can also put a little bit of blurring here if you would have a lot of jagged edges and stuff like that in the brain. That will sort of smooth out the results. So it's always a little bit good with the segmentation of the brain section itself to have a little bit of blurring, but not too much. Like 3 is good. And now we can start looking at segmenting the neurons. So that's the intensity we normally start with setting the intensity range where we have GFP signal in this case. So as you saw from the ImShow command, it's somewhere around 6,000 it starts, and then it sort of ends somewhere 20,000, somewhere there. That's the range where we find neurons. So this is normally a good start to start with. Can already see it looks a little bit nicer. We seem to find the cells, and we don't get like autofluorescence, like dirt on the glass and stuff like that are not shown up as any cells signal. We can also see that certain cells we don't get because they're so close to the autofluorescence, right? By turning on H, you can turn on and off the red signal, which is your segmentation result. So we can also see here that we seem to get a lot of large blobs and also like small autofluorescence here. So we're going to try to reduce that by changing other parameters, for example the Soma size. So the minimum allowed Soma size I'm going to set to sort of like, yeah, it has to be like 3 pixels, larger than 3 pixels. And then you can see we remove this kind of like autofluorescence, or like rather this like processes that also is fluorescent but not the size of a cell body. So here we have two large cell bodies, which obviously cannot be cell bodies. So in order to still get those neurons, but we're just reducing the largest range of a neuron. So we set that to like 100 pixels in area, and you can already see that we still get the neurons, but they are not grouped together like a huge neuron, like a huge cell body. Eccentricity is an option, which is just the max is interesting. Eccentricity basically how spherical or if it's an ellipse shaped cell body. So if it's set at 1000, it means you allow like elliptical shapes object. But if you remove it down, so for example, removing apical dendrites, you saw there that I could remove the apical dendrites from the segmentation and only get the cell bodies. You see here, if I put it up again, I get the apical dendrites. So how elongated in space the shape is, or if it's just more like a perfect dot. Okay, so I mean, I think this is going to be good for now. You can see a lot with this. So I pressed escape, and it then outputs now and shows that we got 811 neurons segmented out in this section here. So in this seg object, we will look what we have here now by the name command, and we have two objects in this list, which is both the filter parameters we just set and also this object called Soma. So let's check at the filter first. So the filter contains both the area limits, 3 and 108 we set it to. Also the threshold range, which is the intensity where you have this fluorescent signal you want to find neurons in. Eccentricity, max and min here is like the 8-bit render where you want to show your result in JPEGs, stuff like that. And then you have the resizing and the blurring here. And brain threshold for segmenting the brain outline. So we can then, for example, change anything manually by coding like this. So now I change the blur from 3 to 4 just by changing output. Now I'm changing it back again. So you can do this loop through, for example, a spreadsheet or anything and have already parameters set as you want. Let's look at the Soma. So this is actually your result that you have segmented now, the cell bodies. So the Soma object contains basic information like the X, Y pixel coordinates of the centroid of the Soma, the intensity in fluorescent intensity average, and the area. All of these results are there for you, for example, to plot as I'm doing now. So now I'm going to plot the position of each neuron in X and Y coordinates in pixels. So if I press there, I get this plot, and you can notice here how it's upside down. The cortex is down and the stratum there with the injection site is up. And this is of course because pixels coordinates actually starts in the top left corner. So I can change that by setting the y-lim, the range of the limits of the y-axis, by reversing the range of my variable there, the y variable, which is this Soma y. And if I set aspect, ASP to 1, no matter if I resize like this, the aspect ratio between the width and the height is going to be the same, which is also kind of nice when you work with this more geometric data. Okay, so let's look at registration now. So for the registration, you could do that without segmentation, of course, but I'm going to show you how to integrate the segmentation results into your registration in order to get where in the brain are these neurons. So in order to do that, I will actually start by opening a plot window because I want a large plot window here. So yeah, we're going to hide that behind here. Okay, so we're going to use the registration command, and we're going to save like we saved the seg, the segmentation command into a seg object. We're going to save the registration output into a regi object. I mean, we could really call it anything. So the registration command here has as input is basically the same as segmentation, your TIFF image, but you also need to provide the coordinate and set it like, for example, coronal here by default. Then you have all these other parameters like brain threshold and resize stuff like that, but we're not going to use them since we already set a filter. So number of nested objects is a little bit interesting here. That's if you have nested like ventricles. It's by default set to 0, but if you, for example, wanted 2 ventricles, you would also set that to 2, for example, if you have 2 ventricles nested within your brain outline contour. So we're inputting the file name, the FFC, the stitch, and I think this coordinate is roughly sort of 0.38 from Bregma. And then as filter, I provide the filter parameters in my segmentation object that I already set. So we're going to use that when we're rendering the output. So now we run the registration here. It's first loading the image, and now it has actually started. So it starts by finding the equivalent like plate in the coronal axis of the WholeBrain atlas, and this we can already see, like, and this seems to fit with what we saw when we were working with the segmentation. So this roughly is where we are in the atlas. And now it's actually done and finished. So it's first left there plots the reference. Basically, this is the WholeBrain atlas, the idealized, and to the right is your fit here now, where it actually estimates that this is where it wants to place. And the way it places this contour is by finding correspondence points, and this is very important in order then to, for example, manually adjust any sort of registration results. You can see here that the point 17 here is a little bit off here. It's not in the midline, and there are other things that we might want to actually do something about. For example, the ventricles. So we're going to start with the ventricles here, how to add correspondence points, because these black dots there were called correspondence points. So we output to Regi object, we use add core points, and it takes an input, our registration object, for example Regi that we just used, and then the number of points you want to add. So we use Regi here, and then I want to add 10 points, 5 for each ventricle. So it starts like begin with defining the core points in the target image to the right, which is the purple here, and then the same equivalent coordinate in the atlas in orange here. So I start there, and I think the same coordinate point is sort of in the atlas roughly here. And then I go to the ventricle here in the image, and it's here, and in the atlas it's here. So it's not in the purple outline I want to mark, rather in the image, whereas in the orange here I want to mark where in the atlas it is, so in the orange outlines. So I'm going just to like make a triangular shape here of the ventricles, and then sort of adding the sides of two sides of the ventricles here. Because they're a little bit larger in my target image. So here, yeah, and that sort of is here in the atlas, and on the other side as well, and here, and we do it for the other ventricle as well. So there, and there, and last one. Yeah, and when you press the last one, you see that the crosshair is gone now, and it has registered. I added 10 points here, 5 by 5, 5 for each ventricle. So now I can actually rerun my registration using these newly added core points, including the correspondence point that we already have. So in order to do that, I just copy and paste the registration command, but I add this line where I use correspondence. And the correspondence is basically your registration object. So I add Regi here, which I added the correspondence points to, and output it to Regi. So now we rerun it here, and we will display the results in this window again. So we just wait for it to finish up. We will start plotting immediately when it has fine. Yeah, so now it's first always to the left here. It plots the atlas looks the same. It hasn't changed. Because we haven't changed the segmentation parameter, but here you will notice a little bit of different in the ventricles here, right? They fits better with the object, and you can see here now we have new correspondence points here, the points that are about 32, 33 to what is it, like 42. Yeah, so we also want to maybe change some correspondence, like 17 there was not in the midline. So if I want to change them, I use the similar output to Regi, but change core points and not add. And this command takes as input similar to add. You use your registration object, but then you provide a vector with the points. And this is the numbers that you saw there, the points that you want to change. But in our case, we want to change sort of 15 to 20, in that range. We want to put them better in order. So we're going to write 15 to 20. And this is actually the order that we want to change them. So it says like change where you want to position point 15 in the target image only. So we start with 15 here. And target image is only the purple here. So we don't click here in the orange at all. So we start 15 here. I think it should be placed there. 16 there, and then 17 in the midline here, clearly, and 18 roughly here, 19, yeah, sort of there, and then 20 there. Okay, so now when I've done that, it's simply rerun the registration again to see the output. So yeah, just comment here that this is a change components. Okay, so we just rerun it and we will look as it produces the output here for us. So okay, there it starts plotting. Okay, you can already see that midline is properly placed now. And I mean, we can of course continue here. For example, the corpus callosum is really a little bit overlapping with the stratum, and also maybe the layering you want to adjust a little bit. But I think this is good for now. Like, I mean, normally if the first time we just did it without any manual adjustment, I think the quantification would also shown like pretty good regardless. So let's look at how you save the segmentation registration. This varies. Just write save, and then the objects, any objects really, you want to save from your working space, and then a file that you want to save it to. So seg and rigid objects, and then we call this file example section.rdata, which is the file format for loading R data. And then we just run this, and now we should have in our default directory, so get working directory. So they should be saved here. So if I go to this folder on my computer here now, I'm going to find this new file here. So I'm going directly like this, yeah. And let's look at the latest saved objects. Okay, no, this is old. Yeah, there we have it. Example section.rdata, and this we can just drag and open in R directly, and it's 2.1 megabyte, and this is the registration and the segmentation results, all that we need in order to actually redo this. Or actually, so we don't have to redo this manually or anything. Everything is now saved there. So the computer can redo this and apply it to another image, for example, or anything really. So let's inspect our results here now. Basically plotting the registration results, and when we do this we can also immediately get sort of like spreadsheet data from where each cell is in the brain, like which brain area, already when we do this inspection, which I sort of highly recommend when you just do this one simple brain section. Maybe not when you run multiple brain sections, then it's other things in order to look at your data if it's accurate or not. So yeah, we saved to an object called dataset. We use the inspect registration command, and it takes as input our registration object and a segmentation object. So we write our registration object to regi, and our segmentation object is seg. And then we just simply run this, and it will output to dataset here. So it actually plots here. So it starts plotting first of all. I mean the atlas section in cryo-section coordinate system in millimeters here, you see. And to right you have your registration results here on the superimposed, and you also see the sort of deformation field in purple, and all the cells here you can notice now that they are color coded according to which brain region they are, so like blue for stratum and green here for cortex. And you can also notice here these red ones. These are actually a warning. Like if you have a black cell with like red outline, it means that these neurons have not been properly assigned to any brain region. And if you look closer, these are probably not neurons, but rather like something that we didn't get intentionally when we did the segmentation. You also see in the left Alan Atlas slice that we don't see any neurons there, and that's because we never applied the forward warp. The forward warp command is default set as true, so we only compute the backward warp, which is basically the transformation from the atlas to the section. we're looking at that later. But now let's look at the data set. So if I press head data set, it just shows me the first 6 rows of the data set, which are the first 6 neurons here. So each row is a neuron, and we can see the first column is the animal, then we have the anterior posterior coordinate, we have the coordinates in X and Y in pixels where the neuron is, the centroid, and also the brain region, both like acronym, name, and which image it belongs to, if it's in the right hemisphere, and stuff like that. So I'm going to show to you now how you would, for example, insert the name of the animal here. So if I subset the data set, the animal here, and I'm going to give the name of this mouse 50 Moscovich. So I write that there as a string, and I insert it into this data set animal, the column there. So by default it's just NA, like not applicable, which is like empty. But now I have inserted it, so we can then look at the data set, and we can see that this single value replaced all the values. All the rows there. So for all the observation, we'll know that this neuron belongs to this animal, this 50 Moscovich. Okay, so let's try to find these observations of the neurons, the red neurons that were outside of the brain. So if I then apply this which, actually no, we don't need a which at all. We can just subset our data set and find the rows, that's the first entry, where the ID is 0, because that's regions that are not assigned. And you see here it gives us these cells here that have not been assigned a region here. So if I then take these observations and I set dataset to be anything that are not, the exclamation mark means not here, then I will remove all the dataset that have 0 ID, where regions do not belong. And you see if I run which on them, it shows up integer 0, which means there is no case that fulfill this logical criteria. Okay, so let's look at the forward warp now. So I just copying the inspect registration and set forward warp to true. And now it will go and basically redo some of the registration. So this will take a little bit of time computing the forward warp. So fast forward here, and now it's plotting then the output with the forward warp. So you will notice what is different in this plot. So you see here now I get the transformation field in the forward warp and the neurons, their position in the stereotactic atlas, which is basically what we need in order to reconstruct the whole brain. Okay, so that's with the forward warp, how you get the transformation from your brain section to the atlas of the single neurons as well. So what we now will look at is actually to plot this data. We want to now quantify this data set, showing it, for example, in a bar graph or something like that, like cell count. So we will make a plot of the results. So there's a command called bar graph, which in this case actually will be a dot graph. So I just apply my data set to this one, and then we'll start plotting it. So here you see all the unique regions, like you see all this, like the name here, and also the acronym, and then you see basically each dot here is a single brain region, which on the x-axis we have the cell count in that brain region. So filled circles here is like regions that are located in the right hemisphere, and the hollow circles are the left hemisphere. So we can see contra versus ipsilateral input here and see interesting patterns. Yeah. No, what we want to do is we want to save this plot now. So in order to save it, we can save the result to a file. I will just name this, for example, contra versus ipsi example section.pdf, and then you write type equals to PDF, but you can also write PNG if you want a PNG result. But these PDF are really nice. They can be opened in Illustrator or Inkscape, whatever you prefer for making publication quality figures, and we can search on it or like rather see where it's saved. It's saved here by default, your working directory, which for me now is this the user default pathway. So I just go there and find it where the file is saved, and here you see I saved as a PDF this controversy ipsilateral, and if I open this up, you see a PDF file with the plot here, which is kind of nice. And this of course now can be open in Inkscape or Illustrator in order if you want to put it in as a panel in a larger figure. Let's also save the figure from the inspector registration. I will just remove the forward warp so we don't compute that again and remake the plot. So I want to save this just to show you that this can also be saved as a PDF plot with all the polygons and everything in vector format, so you can easily change any color or anything you want in Inkscape or Illustrator manually afterwards, or insert it in a larger figure as I said. So now when it's plotted, I just copy this, and it will by default save the active plotting window, the latest active plotting window. So inspect registration, we should now have a file in my working directory, which is there, and here it is. And if I open this one, you can see that you have a PDF here. So I close both of them. So the last thing I'm going to show is just one purpose of actually having image processing in R, because it's opened up all the possibilities of using the statistical advantage that R has. And I mean, the most simple example is just creating frequency tables. So I'm going to create a frequency table of the number of brain regions, or like cells in brain regions. Here you see it. This is just a table here. Let's run it again. There, where we see, for example, anterior cingulate cortex, dorsal layer 5, cordatum putamen 517. So these are the number of cells detected in these regions. We can then do cross-tabulation by, for example, looking at the different hemispheres, and this is basically how the plot is produced that you saw previously. True here means it's in the right hemisphere, and false means that it's in the left hemisphere. So we have two columns, and each row is then the brain region. And we can output this into a new object that we call do some stats on. For example, if you want to apply all the different statistical techniques that other packages in R might provide. So in the two upcoming videos, we will look, first we will going to look how we can extend this and basically do processing of multiple image sections in a pipeline, like batch processing for an entire brain, several hundreds of sections. And then I will also make a separate tutorial on how to render output for the web, basically making your own sort of like neuroanatomical maps that you can share online. But this is it for now.